This is part 3 of our sample prep presentation. We're going to talk a little bit about cell cultures, paraffin sections, and antibody staining. In most cases, you're going to need an inverted light microscope to look at cell cultures. Plastic culture dishes are really only useful with phase contrast imaging, just to check on your cells to make sure that they're doing all right. They're terrible for taking high quality images. And if you're trying to do differential interference contrast imaging, which is another technique for looking at transparent cells, they simply won't work in plastic dishes at all because DIC uses polarization to create the images, and plastic is a polarizer. If you work with optical microscopes, you probably heard this before, that most of our lenses expect a number 1.5 thickness cover slip. And that thin piece of glass is actually factored into the optical calculations when the lens is designed. The wrong thickness will produce aberrations or distortions in the image, and the higher magnification / numerical aperture of the lens, the more that becomes a critical issue. You can see that from the graph over there on the right. And you'll see this at the bottom of, I think, every slide in this portion of the presentation, that everything that interacts with the light in an optical microscope affects the image quality. This includes the sample, which you have the most control over, the mounting and culture media, the dish and the cover slip, the air, oil, or water between the lens and the sample, and the quality of the microscope itself. Of all that, the core facility primarily can only control the quality of the microscope. The rest of it is on the user. Now, some cell types don't grow well on glass. We've discovered that. We've seen when we've been imaging, the cells just kind of lift off. So if you can coat it with some sort of protein like fibronectin, polydilysine, collagen, that will help your cells grow on the glass surface. And you can buy the dishes. Some people make them, but you can buy the dishes. They're relatively inexpensive, and you want to make sure that you get the correct cover slip thickness, because incorrect cover slip thicknesses don't help you either. If you have especially difficult to grow to cells, you might want to investigate culture dishes from a company called Ibidi. They have a proprietary plastic that they say is similar to the refractive index of a number 1.5 glass cover slip, but it's much more culture friendly than glass. Now, we've worked with these a little bit, and let's just put a little caveat on there that similar is nice, but it's not glass. And we have seen that the proprietary plastic sometimes reacts poorly with the hydrocarbons in the immersion oils, and they do have a list on their website of oils that cause issues. Sometimes the oil clouds it up, especially if you're looking for a long period of time. Now, there are some other technical challenges with live cell imaging, and this is where having an experienced core manager to interact with is very useful. You want those cells on your cover slip, not on the surface of a microscope slide. Now, we sometimes see these types of chamber slides. You notice the blue resist that's on there to hold the plastic chamber to the actual slide itself. Our experience has been that the images from these type of slides are lower resolution, and they are considerably dimmer when you compare them to images of cells that are grown directly on the cover slip. I know they're less expensive, but use cover slip bottom dishes. There are also chambers like this that have a cover slip bottom. Use those instead. These lead to terrible images. And here's why. So if you look at this, you can see the thickness of the microscope slide. You have that blue resist or paint, whatever you want to call it. Up here you have the cover glass, and you have some thickness of mounting media. So let's figure that out. Okay, so the thickness of a microscope slide is about 1 millimeter. We know the thickness of a 1.5 cover slip is approximately 0.17 millimeters. And this is just a guess. We'll call this distance about 0.3 millimeters from the microscope slide to the cover glass. If you're trying to look with an inverted microscope through a microscope slide, you're looking through an enormous amount of glass that was not factored into the calculations when the lens was made. And you will find that your cells are far away from optimal. And with cells being small, that really creates a problem. They start to distort. Now, if you can look at them from a side where you can get a little closer, a 10x lens is going to have no trouble seeing your sample, but your 10x lens, which focuses quite far away from the lens itself, does not have the resolution that most people want. As you go up to a higher magnification lens, the focal point, or what they call the fixed working distance, now becomes an issue because it will only go a certain distance away from the front of the lens. And if your sample is a long way away from the front of the lens, we can't focus any closer. And the differences in refractive index between the glass and the mounting media, which is typically glycerin-based, can cause aberrations, and we're going to talk a little bit about these a little further on. But you have to have mounting media between that blue resist and the cover slip, so that creates even more distance from that the light from the cells has to pass through, and it's not a particularly good refractive index match. Paraffin embedding requires you to take a sample that has water in it and dehydrate it. So typically you'll go through a series of changes of ethanols from 70% to 100, and then you'll do some changes of xylene to remove all the water. And now it's in a solvent that's miscible with the paraffin wax that you can get it into the sample. The paraffin is typically at about 50 to 60 degrees C. At this point, because of the xylene, most of the lipids have leached out. So if you're looking at fatty liver, for example, you'll have holes where the fat was. You can process samples by hand in a lab, but these automated vacuum tissue processors that histology core facilities typically use will greatly improve your sample quality. Even if you only use them to embed the blocks, you will get better blocks. And here at the University of Arizona, there are 2 histology core facilities, one in the pathology department and one in the cancer center. Paraffin sectioning can be done by just about anyone with access to the appropriate equipment, but someone who does this full time is typically going to produce better sections and give you better quality staining. If you happen to have access to a microtome and you're going to do the sectioning, I encourage you to respect the microtome as a dangerous tool. I managed a core facility for almost 20 years, and we had someone take the tip of their thumb off with a microtome. If you're sectioning tissue, we strongly encourage you to use charge slides. Again, a story from the past. We had a student cut through some tissue and made tons of slides using standard uncharged slides, and was in a panic when the sections all floated away when she tried to do immunostaining. The problem was that we couldn't fix it after the fact. Charged slides are your friend as far as keeping those sections attached. And paraffin sections can be from about 4 to 10 microns. 10's about the most you can do, and 3.5 to 4 is about as thin as you can cut it. And again, if you need it really thin, you probably need a professional to be cutting those sections. And there are many excellent stains for histology, including special stains that will show you different things about the tissue, and even enzymatic assays that work very well. You don't have to use immunostaining. Now, primary antibodies that work well on paraffin sections, you need to read the instructions and make sure that they're not just for frozen tissue or for westerns, because they're not, if they aren't specific that they will work with fixed paraffin embedded tissue, they may not work very well or work at all. And the truth is, out there are some vendors that have better reputations than others about the quality of their antibodies. I have a friend who was involved in QCing antibodies, and he said sometimes there's 20 different antibodies that you can find in the literature against particular proteins, but some are much more reproducible than others. So pay attention to that. Don't just grab the first thing you see, but make sure that it's something that has an excellent reputation. Newer antibodies may even have a research resource identifier or an RRID. You can look for that. Test it. Do positive and negative controls. If it's for actin, does it stain muscle tissue? Make sure you're using appropriate lot numbers. If you have to change lots in the middle of an experiment, you want to rerun your controls and make sure that what you're getting with polyclonals in particular actually works the second time. I know of a faculty member that the rabbit that they were making her polyclonals in passed away, and the new was totally different and did not stain things as expected. And this applies to immunohistochemistry using DAB staining or immunofluorescence or even in situ hybridization. You may see discussions about antigen retrieval. It's actually a collection of techniques often using a microwave or buffers and different types of buffers to heat the sample to reverse the cross-linking of formaldehyde. There is no standard antigen retrieval technique. Different types of buffers work better with different tissues and different pHs. You need to know how to use a microwave to prevent overheating or denaturing the sample. You want to do it consistently. It's not my area of expertise, but there are books about doing microwave antigen retrieval. And you will want to test your protocol and make sure that you get the best results consistently. And here again are our image sources.